Okay, ladies and gentlemen, we're back. Today we're going to look at this human karyotype packet that you have. It's a fairly big one, and we're going to go through some additional notes to add to it, which will help you with your test coming up on April 7th, as well as some other questions that we have. So a little bit of background on this is that karyotypes are a way that we classify chromosomes. Karyo, the prefix means nucleus. It came out in about 1966. You have to remember that way back in time in the early 60s, no one really had an idea of what was going on genetically. Then people started to go, oh, look, that family resembles each other. Why do they resemble each other? What's going on here? And we finally come up with this idea of chromosomes and come up with the structure of DNA and start to realize all the things that you guys get to learn about today. So you certainly can read this on your own, but some things you should be familiar with. So our chromosome is here, and this would be our sister chromatid pair that is attached at the centromere, the point of attachment. Where they attach, you're going to have a short arm and a long arm. The short is the P arm and the long arm is the Q arm. And then there are numbers. They're not really there, but we number from the centromere away. And you'll see that in the next page a little bit to help people to identify and understand what part of a chromosome are you talking about? So that if I think that my patient has a disorder that is here, I could identify this as chromosome 1, Q2, and then further designate an actual location on the chromosome where I think that there is an issue. So again, P arms and Q arms. The petite arm is the short arm, Q is the long arm. The numbering system helps people to identify. The system we use is called the Denver system. So chromosomes are lined up by size from largest to smallest, and then they are redesignated as letters A through G. A chromosomes are 1 through 3, B are 4 and 5, C are 6 through 12. Again, you'll see this on the back side in a moment. We are looking first at the autosomal chromosomes. So autosomes are body cells, and we are looking at chromosomes number 1 through 22. Number 1 is the biggest or longest, 22 is the smallest. Actually, there should be a flip between the 21st and 22nd chromosome, but they're so bitsy tiny that when people were doing this by hand, it was hard to see. We have chosen not to flip it and make it the way it should be because chromosome 21, Down syndrome, trisomy 21, is a common disorder and it would just kind of confuse everyone. So we've left them with the original system. We talked the other day about amniocentesis. This would be a way of looking at chromosomes to see if there are any genetic disorders based on chromosome number, too many, too few, if something has flip-flopped in them. So when we are writing out our chromosome information, again, this is so you can communicate with people. If I am a normal female, I have 46 chromosomes, 23 from mom, 23 from dad. I got an X from my mom, I got an X from my dad, making me a female. A normal male would be 46 XY, X from mom, Y from dad. If a female is missing a chromosome, they would be written as 45. So they're missing an X chromosome in this case. And this individual would have Turner syndrome. She would be a Turner syndrome female. If a female has an extra X, she would be 47, triple X, or super female. Then we can even designate further. So you're going to see this again, so please become familiar with it. It is our method of helping to give information to other scientists that are looking at chromosomes. So if I have a male that has an extra G group chromosome, it would be written as 47, an extra chromosome, XY, a male, plus a G group. Male with a missing G group would be 45. They're missing a chromosome. They're XY because they are male minus a G group chromosome. So this is going to become important to us as we start to improve our background in genetics. A plus or minus symbol in front means that plus you have an extra, minus you have a missing piece or a missing chromosome. Then we can look at the arms of the chromosomes. So a P minus would mean there's a short arm deletion. There's a piece of the chromosome missing from the short arm. A Q minus, a long arm deletion. One of our more common genetic disorders that we talk about and designate is called Karyotype syndrome. It is also known as cry of the cat. These babies have a very shrill, shrieky, crying sound. You can look up on YouTube and watch a baby crying that has Karyotype syndrome. People that have this syndrome, that is probably one of the first characteristics of the disorder. They are 46 chromosomes, so they would appear as normal, XX or XY, so this would be a male, and then B, P minus, missing a part of a B chromosome on the petite arm. On the long arm, it would be 46XY, BQ minus. The B chromosomes are chromosomes 4 and 5, which we covered up here. So if you are a normal male, you would be 46 chromosomes, XY. A B group chromosome is chromosome 4 or 5. Karyotype happens to happen on chromosome number 5, and it would be a missing part of the short arm. So if you saw this designation, P, this is the description you would give of it. Again, you're going to see it again. Makes a little more sense when we get to the next page. You can certainly read about the banding and how originally we came up with these staining techniques and this chromosome banding. The staining techniques have improved tremendously. We also now have a type of banding called spectral karyotyping, which gives us fluorescent bands, which makes it way easier to see and identify if there's a problem with a chromosome. Your next page. So we have different types of abnormalities that can happen. Some are known as whole chromosome abnormalities. We want a disomy. This is good. It is a normal number of chromosomes for a species. In humans, it would be 46, one from mom, one from dad. So here would be chromosome 1 from mom, chromosome 1 from dad. It gives us 2 chromosomes. And again, there would be 23 pairs, giving us a total of 46. A monosomy is when we are missing a chromosome. During meiosis, the chromosomes didn't separate properly, so as a result, we're missing. The opposite of this would be an extra chromosome. 3 would be tri, and tetrasomies can also happen. This would be 4 of the same chromosomes. Aneuploidy is not good because it's different than the normal number of chromosomes, meaning a genetic disorder or genetic dysfunction. And these often happen because of non-disjunctions. The chromosomes don't split properly during meiosis. Other types of chromosomal abnormalities that can occur are deletions, so missing a piece during crossing over, typically in prophase 1, or during the separation of chromosomes if a chromosome gets tangled up. There's a missing piece. That missing piece is going to result in a chromosomal disorder and a genetic disorder, or can. Duplication, extra piece. So a piece is added on that is a repeat of what's already there. We also have translocations. These are when a piece from one chromosome breaks off and it joins another chromosome. So although you have an extra piece of 21st, it has joined the 12th chromosome. An inversion, a flip-flop of genetic information. The sequence should be the way it is. It has flipped. So again, you could see how that could lead to mutations if in fact there is a problem with an individual. So this shows us our 23 chromosomes, and chromosome 1 is the largest, 2 gets smaller, 3 gets smaller, they get smaller, 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 until you're at your 22nd chromosome. A group chromosome, B group, C group, D group, E, F, G, and then off to the side you have your sex chromosomes. So X chromosomes are fairly large, they would fit in the C group category, and Y chromosomes are very small, they would fit in the G group category. Y chromosomes, if you have one, you are a male. If you do not have one, you are a female. So that's going to wrap up this beginning portion of our karyotype packet. We'll pick up a little later, actually, with the disorders and talk about them a little bit more. Have a good day.